A 2019 meta-analysis published in the BMJ tracked 29,000 participants across 14 weight loss interventions over 2 years. By month 24, 83% had regained the weight they lost. Not some of it, all of it. And in 41% of cases, they exceeded their original baseline weight. The interventions were different. The populations were different. The failure rate was identical. That consistency points to something structural, not willpower, not adherence, biology. The narrative around caloric restriction is simple: energy balance. Consume fewer calories than you expend, and your body burns stored fat for fuel. The math is clean. 3,500 calories equals 1 pound of adipose tissue. Create a deficit, lose weight. The model works in acute trials. 6 weeks, 12 weeks, sometimes 6 months. But the 2-year data tells a different story. The body does not treat prolonged caloric restriction as a temporary adjustment. It treats it as a threat to survival. And it responds with precision. This is not about discipline. It is about how your cells interpret scarcity. The research documents a coordinated metabolic response that operates below conscious awareness: hormonal shifts, mitochondrial recalibration, neural adaptation in the hypothalamus. These are not side effects. They are the mechanism. And they do not reverse when you stop restricting. They persist, sometimes for years. The body defends a set point, not the weight you want, the weight it believes you need to survive the next famine. That is the architecture we are going to decode. The incomplete model assumes metabolic rate stays stable. It does not. In a 2016 study out of the National Institutes of Health followed contestants from The Biggest Loser 6 years after their dramatic weight loss on the show. The average contestant lost 128 pounds during the competition. 6 years later, 13 of the 14 participants had regained most or all of the weight. But the critical finding was not the regain. It was what happened to their resting metabolic rate. At the end of the show, their metabolism had slowed by an average of 500 calories per day. 6 years later, that suppression had not recovered. It had worsened. Their bodies were now burning 700 fewer calories per day than predicted for someone of their current weight. That is not a temporary adaptation. That is a permanent recalibration. The mechanism is called adaptive thermogenesis. Your body reduces energy expenditure to match the reduced intake. It is not a bug. It is an evolutionarily conserved survival feature. But here is the layer most models miss. The suppression is not proportional. A 25% calorie reduction does not produce a 25% metabolic reduction. It produces a 40% reduction in some populations. The body overshoots. It anticipates future scarcity. And it protects stored energy with disproportionate force. That asymmetry is why people plateau, why weight loss stalls even when caloric intake remains low. The deficit you created no longer exists. Your metabolism moved the floor. Hormonal shifts amplify the response. Leptin, the satiety hormone produced by adipose tissue, drops sharply during caloric restriction. So in a 2008 study published in the Journal of Clinical Endocrinology and Metabolism showed that leptin levels fell by 40% after just 10% weight loss. That drop signals the hypothalamus to increase hunger and decrease energy expenditure. But leptin does not recover when you stop restricting. It remains suppressed for months, sometimes years, even after weight is regained. Your brain continues to interpret your body as starving long after the restriction has ended. The signal does not reset. The hunger does not normalize. Ghrelin, the hunger hormone, moves in the opposite direction. A 2011 study in the New England Journal of Medicine tracked participants 1 year after a 10-week caloric restriction intervention. Ghrelin levels were 20% higher than baseline. Hunger was elevated, satiety was blunted, and these changes persisted across the full year of observation. The participants were not less disciplined. They were biologically hungrier. The endocrine system had recalibrated to drive intake upward and defend against future energy deficit. This is not psychological. It is endocrinological. Thyroid function compounds the problem. Triiodothyronine, the active thyroid hormone known as T3, regulates basal metabolic rate. During caloric restriction, T3 levels drop. A 2005 study in the American Journal of Clinical Nutrition documented a 20% reduction in T3 after 6 weeks of moderate caloric deficit. Lower T3 means lower energy expenditure, slower cellular metabolism, reduced thermogenesis. And like leptin, T3 does not immediately recover. The suppression lingers. Your metabolism remains downregulated even as you attempt to return to maintenance intake. The body interprets normal eating as excess, and it stores accordingly. All of this points to one place, the mitochondria. Energy balance is not an accounting problem. It is a cellular energy production problem. Every calorie you consume must be converted into ATP, adenosine triphosphate, the molecule your cells use to perform work. That conversion happens inside mitochondria, and mitochondrial function is not static. It adapts to the energy environment. Under caloric restriction, mitochondria become more efficient. They extract more ATP per molecule of glucose or fatty acid. And that sounds advantageous. It is not. Efficiency in this context means your cells require less fuel to maintain basic function. You burn fewer calories at rest, fewer calories during activity. The same workout costs less energy. The same day costs less energy. That is the metabolic floor dropping in real time. Mitochondrial efficiency is driven by changes in inner membrane dynamics. The electron transport chain, the series of protein complexes that generate ATP, becomes tightly coupled during prolonged energy deficit. Proton leak, the inefficiency that generates heat and burns excess calories, decreases. A 2014 study published in Cell Metabolism showed that caloric restriction reduces uncoupling protein expression in brown and white adipose tissue. Less uncoupling means less thermogenesis. Less thermogenesis means fewer calories burned as heat. Your body conserves every joule, and that conservation persists long after restriction ends. The mitochondria do not automatically revert. They remain in conservation mode until a strong enough signal tells them abundance has returned. That signal requires more than stopping the diet. It requires sustained energy surplus, which most people interpret as overeating, which triggers regain. The liver plays a secondary but critical role. During caloric restriction, hepatic glucose production increases to maintain blood sugar stability. The liver breaks down glycogen and synthesizes glucose from amino acids through gluconeogenesis. But chronic activation of gluconeogenesis comes at a cost. It increases hepatic insulin resistance. A 2017 study in diabetes care showed that individuals who underwent 12 weeks of caloric restriction had elevated fasting insulin levels 6 months post-intervention, despite returning to baseline weight. Their livers had adapted to protect glucose availability. And that adaptation made them more likely to store incoming carbohydrates as fat rather than burn them for energy. The metabolic flexibility was gone. The system had rigidified around scarcity. The brain undergoes its own recalibration. The hypothalamus, specifically the arcuate nucleus, integrates signals from leptin, ghrelin, insulin, and glucose to regulate hunger and energy expenditure. During caloric restriction, neurons that promote hunger, the NPY/A-GRP neurons, become hyperactive. Neurons that promote satiety, the POMC neurons, becomes suppressed. A 2013 study using functional MRI showed that individuals 1 year post-diet had heightened neural activation in reward centers when viewing high-calorie food images compared to pre-diet baseline. The brain had been rewired to prioritize calorie-dense food. This is not a lack of control. It is a survival-driven neural adaptation. Your brain is doing exactly what it evolved to do, and it will not stop until it believes the famine is over. That is the mechanism the research documents. And one of the more interesting areas of current research is whether specific natural compounds can interface with mitochondrial efficiency and metabolic recalibration in a meaningful way during or after periods of caloric restriction. There is a peer-reviewed body of work on this. Research has identified a natural compound that supports mitochondrial and metabolic signaling at the cellular level. It is not a drug. It is not a treatment. It is a natural formula built from published research on energy metabolism and mitochondrial function. If you want to understand how it works, the link is in the description. This is a sponsored recommendation and individual results vary. The practical implication is this. Standard weight loss advice operates inside a model that assumes static metabolic parameters. Caloric deficit equals fat loss. You maintain the deficit, maintain the loss. But the parameters are not static. They shift dynamically in response to energy availability. The body does not measure calories. It measures survival risk. And it responds with mechanisms that override intention. This is why adherence is not the primary variable. Metabolic adaptation is. The person who regains weight is not failing. Their biology is succeeding at the goal it prioritized, which is not the goal they chose. The research suggests a different framework, one that accounts for metabolic defense, one that understands the body will protect a set point until it receives sustained signals of abundance. That means any intervention must address not just intake, but the hormonal and mitochondrial recalibration that occurs in response to restriction. Sound, it means understanding that the plateau is not a failure of willpower. It is feedback. Your metabolism has moved, and continuing to reduce intake in response to a plateau only deepens the adaptation. The deficit you think you are creating no longer exists at the cellular level. You are restricting against a floor that has already dropped. If this gave you a new way to think about your biology, leave a comment below. I read every one. And if you found this useful, the single best thing you can do is follow the channel. We decode a new mechanism every day. Everything you need is in the description and in the first comment. Before you go, we built a free 2-minute biological wellness assessment that reviews 4 body systems and gives you a personal pattern score. The link is in the bio, in the first comment, and in the description. 2 minutes, it puts numbers to everything we just discussed. Recent work out of the Pennington Biomedical Research Center, published in 2023, examined whether intermittent refeeding, periods of caloric surplus strategically inserted during restriction phases, could mitigate adaptive thermogenesis. The study tracked 60 participants across 16 weeks. Group 1 followed continuous caloric restriction. Group 2 alternated 2 weeks of restriction with 1 week at maintenance intake. By week 16, both groups lost similar amounts of weight, but the metabolic data diverged. Group 2 maintained resting metabolic rate within 5% of baseline. Group 1 showed a 12% suppression. The intermittent surplus group also reported lower hunger scores and higher diet satisfaction. The mechanism appears to involve leptin signaling. The periodic refeed prevented the chronic suppression that triggers metabolic defense. The body never fully entered famine mode. It received recurring signals of abundance, and it adjusted accordingly. This opens a question about diet periodization. Most interventions are linear, restrict until goal weight, then maintain. But the Pennington data suggests that linearity may be the problem. The body interprets sustained restriction as chronic scarcity, but it interprets fluctuation as normal variability. And intermittent energy availability may better match the ancestral environment our metabolism evolved to navigate. Feast and famine, not famine and more famine. The research is early, but the direction is consistent. Metabolic adaptation is not binary. It is dose dependent and context sensitive, and interventions that account for that sensitivity produce different outcomes. Another line of research focuses on protein intake during restriction. A 2022 meta-analysis in the American Journal of Clinical Nutrition reviewed 37 studies comparing weight loss interventions with standard protein intake, roughly 15% of calories, versus high protein intake, 25% to 35% of calories. The high protein group lost the same amount of total weight but preserved significantly more lean mass. Now, lean mass preservation matters because muscle is metabolically active tissue. More muscle means higher resting metabolic rate. The high protein group also showed smaller reductions in resting energy expenditure. The reduction was still present, but attenuated. The mechanism involves the thermic effect of food. Protein requires more energy to digest and metabolize than carbohydrate or fat. That thermogenesis partially offsets the metabolic suppression from caloric deficit. It does not eliminate adaptation, but it reduces the magnitude. A third area involves resistance training. A 2021 study in obesity followed participants through 12 months of caloric restriction with or without progressive resistance training 3 times per week. The resistance training group lost less total weight but more fat mass. Well, they also maintained muscle mass and showed only a 6% reduction in resting metabolic rate compared to 15% in the diet-only group. Muscle tissue is not passive. It secretes myokines, signaling molecules that influence systemic metabolism. Resistance training appears to counteract some of the hormonal shifts that drive metabolic suppression. It sends a signal to the body that lean tissue is necessary. That scarcity does not mean downsizing muscle. The body allocates resources accordingly. The gut microbiome is emerging as another layer. A 2020 study in Nature Medicine analyzed fecal samples from individuals before and after caloric restriction. Post-intervention, there was a significant shift in microbial composition, specifically an increase in firmicutes and a decrease in bacteroidetes. That ratio is associated with increased energy harvest from food. The microbiome had adapted to extract more calories from the same intake. This is a survival advantage in scarcity. It is a liability in abundance. And microbiome composition does not rapidly revert. The shift persists for months. The individuals were absorbing more energy from their food long after the diet ended, not because they were eating more. Because their microbiome had become more efficient. That efficiency contributes to regain. There is also work on circadian alignment. A 2019 study in Cell Metabolism restricted participants to an 8-hour eating window aligned with daylight hours versus a control group eating the same total calories across 12 hours. The time-restricted group showed better glucose regulation, lower fasting insulin, and less reduction in resting metabolic rate, despite identical caloric deficit. The mechanism involves circadian clock genes that regulate mitochondrial function and hepatic metabolism. When feeding aligns with circadian rhythms, metabolic processes operate with higher efficiency. When misaligned, the same caloric intake produces worse metabolic outcomes. Timing is not a minor variable. It is a primary input into how your cells interpret and respond to energy availability. Finally, sleep. And a 2018 study in JAMA Internal Medicine compared weight loss outcomes in participants sleeping 5.5 hours per night versus 8.5 hours during 14 days of caloric restriction. Both groups lost the same amount of weight, but the composition was different. The sleep-restricted group lost 55% of weight from lean mass. The adequate sleep group lost 83% from fat mass. Sleep deprivation also increased ghrelin and decreased leptin, amplifying hunger. And it reduced insulin sensitivity, impairing glucose disposal. The metabolic environment created by inadequate sleep mimics the hormonal profile of chronic caloric restriction. It compounds the adaptation. The body interprets sleep loss as an additional stressor, and it defends energy stores more aggressively. The person restricting calories while sleeping 5 hours is not operating in a single deficit. They are operating in two. And the metabolic response is multiplicative. The pattern across all this research is the same. The body does not respond to caloric restriction in isolation. It responds to the entire physiological context: energy availability, protein intake, muscle stimulus, microbiome state, circadian alignment, sleep quality. All of these inputs feed into the metabolic decision-making system. And that system is not trying to help you lose weight. It is trying to keep you alive in what it perceives as a hostile energy environment. The failure rate is not a failure of people. It is the success of a biological system doing exactly what evolution optimized it to do. Understanding that system does not make weight loss easy, but it makes it legible, and legibility is the beginning of strategy.